Good morning, everyone. It's a pleasure to be here. This is my second association with the group, and it's really nice to have a lot of people join you this morning. I'm going to spend the next 30 minutes sharing my thoughts on the evolution of cancer genomics, where is the world heading, and what's been our experience over the last 6-7 years as a group within that domain. As we've all experienced, and we're doing that today virtually this morning, that the pandemic has raised significant challenges to the healthcare system as a whole. Clearly, that bit of impact has not left the oncology ecosystem either. And the pandemic has created a large number of headwinds for the oncology community, particularly with regards to some of the long-term sequences, some of which we are not aware of today, and we will find out in the coming months and years. There's been a challenge for cancer patients with regards to frequent visits to the healthcare provider for their evaluation, for their ongoing therapies, for monitoring. And we've already seen a significant reduction in the number of patient visits that have taken place, as well as treatment volumes, though they are improving from the height of the pandemic. That being said, the oncology fraternity and clinicians as a whole have shown tremendous flexibility with regards to rapid adoptions of telemedicine, telehealthcare. I think thoughtful changes in therapeutic dosing and scheduling, so we can limit the inpatient infusion visits if we can, and also to try and impact the access of outcomes. But that will really be known, I think, in the coming few weeks and months, as I was mentioning. So increasingly, we've seen, as I was mentioning, an impact in terms of patient care and visits to the healthcare facilities. And despite major advances that have taken place over the last few decades, we still face challenges across the cancer ecosystem. Number of questions still remain unanswered, while we've evolved significantly on our drive towards personalized therapeutic care, especially in the domain of oncology. And I'll talk to you over the next 25 minutes on how that landscape is evolving. How have liquid biopsies started to transform some of those action points? What's been the impact of the COVID from the pandemic standpoint, and where are we really heading with the personalization? Number of complexities still arise and still exist with regards to the activities of cancer in terms of not just cancer propagation, but really with regards to response of therapies. Why do similar cancers of the same organ system behave so differently with different medications, different treatment plans? Why is it that some cancers are highly aggressive, where others are slow when we're looking at similar subtypes? What really propagates the disease, and what can hasten the spread, and which future strategies have a role to play within that structure? And I think we've really seen, as things have moved and things have evolved over the last few decades, we're clearly moving away now from the one-size-fits-all approach. And there's a lot of focus of looking at precision medicine across the genomic variants, and really looking at the use of therapeutics that are expected to confer benefit to subsets of patients where cancer displays very specific molecular or cellular features and subtypes. And this is what we're defining now as the use of precision medicine within that space. And increasingly now we're seeing that genomic medicine, genomic testing has become an integral part of routine oncology, plays a critical role with regards to not just theranostics and treatment selections and decision-making, but also in prognosis and interplay with regards to various assessments that are taking place in that period of time. So this is an excellent overview that was given by Falso a couple of years back, which just basically shows what have been the major changes that have taken place across the various ecosystems in terms of timelines and domains of cancer. And as we know that cancer is driven by mutations, and the aim of identifying helps the clinician to determine which possible targets or treatment options, which were initially thought to be unavailable, can now be used in that space. But truly for the personalization, especially in therapeutics, whether it's chemotherapy or targeted therapy or even checkpoint inhibitors, as you can see from the slide, is really to ensure that these technologies become easily accessible. They're minimally invasive as far as possible, and they follow the molecular makeup of the tumor over a longer period of time in terms of the longitudinal aspect of follow-up. Clearly, we understand there are credible challenges to this process, not in the least tumor heterogeneity and new mutations that come up, including resistance to therapies. And we really need to identify targets that can outsmart the quirks of the DNA and the genomic pathways within that perspective to make a significant change within that space. So just to show you just a broader overview of how things are evolving within the space, quintessentially, cancer is a complex disease. And as technologies evolve each day, and we're far better at it today than we were a couple of years back, we still need to understand and learn more about the different layers of the biological elements that exist and contribute not just to the emergence, but also to the development of the cancer. And typically in those, we've always been looking at the three main pillars, which has been the clinical information that we need from the patient. What is the human biosample information that we're looking at? Is it classically a tumor tissue? Are we looking at various subsets of body fluids? Are we looking at blood, CSF, cytic fluid, pleural fluid? And then really, what is the type of the biomarker information that we really need as part of this process? And that, I think, is quite critical to this. So this just shows you in terms of what's really the interplay of the tumor environment with the with the environmental factors, and what are the signaling pathways from the metabolome to proteins to the epigenetics, looking at expression. So it is a complex process, and in my view, trying to identify only one small aspect, whether it's purely looking at a exosomal driven DNA based approach, in my view, can never clearly provide us enough information that we need as part of that process. And on the right, this is just a depiction of what I call as the cancer immunogram from a recent paper, which just shows you how there are different elements in play, from just the tumor load to the general immune status to infiltration from the T cells, and really the interplay taking place with the interleukins and the checkpoint inhibitors that further drive the complexity and our understanding within this process. So just to showcase winds of change, not necessarily that I'm a Scorpions fan, but it's really to define that a couple of years ago when we started our journey looking at the multiple analytes, everybody came back and looked at reviewing, it's the focus of the direction of what you need from the cancer biome that you need to understand. And as you can see, significantly, when technology evolves, it takes a number of those technical aspects away from that process. And it's important to understand that once a new technology rolls over you, if you're not part of the steamroller, then you're part of the road. And for that to happen, when it's new technology, new science within the space, the science needs to be credible, it needs to be ethical, it needs to be reproducible. And this really just shows you in terms of how we've evolved over a period of time where mobile technology, social media technology has completely taken over where we were a couple of years ago within the space of where we are standing. And again, as we're moving back into the future of this perspective, again, the exponential timeline it's taking for us to move significantly in those next steps as part of the process in terms of surpassing, whether it's brain power or looking at equivalents to how things are evolving. Not necessarily to give you the example of the two cars, but this is where we are in 30 years, where the first car in the 1980s came in, was approved for passenger travel. We're using airbags to a standard of where we are today in terms of using not just at the high or even at the entry level of vehicles that have come through in that space. So that's how science evolves rapidly within the structure in that process. But coming back from the automobiles back to our genomics, we know that cancer is a disease of the genes. We know the heterogeneity and new mutations are a challenge. Since the time that Mandel and Mateus told us about the cell-free DNA, since we've been looking at CTC enumeration associated with progression-free survival for breast, prostate, or colon cancer, the science and the technologies have still remained complex within the space. Traditionally, when we've started to look at various aspects of the molecular insight, we've looked at tissue, we've looked at blood. Challenges exist in terms of looking at standard biopsies, especially when we're dealing with difficult aspects of anatomy, when there are comorbidities, when we're looking at a longitudinal follow-up, and a reproducibility in terms of repeating biopsies remains a challenge, and aligning the patient to do that. And obviously there's been an evolution in terms of looking at the liquid biopsies in that space. where we've got aspects of not just blood, but looking at CSF, sputum, various different body fluids, and to try and get a very quick, comprehensive analysis, which can easily be obtained in a minimally invasive way within that structure and process. That obviously comes with its challenges. You're talking about each milliliter of blood, which has got about 50 quadrillion red blood cells and a handful of circulating tumor cells within that space. So it's really trying to understand the origin of those different sub-elements that we're looking at in this space. And it's not so much as trying to pluck a needle from a haystack, but a needle from a barn in terms of sometimes how those complexities can be driven. Liquid biopsies have been starting to be used for molecular characterization of tumors. As I mentioned, the non-invasive nature clearly helps in terms of repeat sampling. If you're going to be monitoring genetic disease change over a period of time without the need for a physical biopsy. And we've clearly seen increasing evolution of the usage of the biomarkers over a period of time to have biomarkers available for the clinicians at early onset within some of those structures. When you look at the different analytes that are in play, we've looked at circulating tumor cells. I'll talk to you a little bit about our experience with using the new evolution of tumor cells from a cluster standpoint. And traditionally, there's a lot of work that's happening just on the DNA as well as on the exosome as well. Exosomes clearly and RNA clearly of interest to us as well as a group, purely because they're driving a lot of the inflammatory stromal and systemic response changes, as well as deep down analytics of the RNA within that structure. Excellent talk from Professor Tor. He talked more about the immunohistochemistry in terms of how that's evolving. And in addition to that, we've been looking at the DNA and the RNA through various aspects of sequencing ourselves as an organization, trying to identify different specific mutations, copy numbers, different epigenetic expression pathway changes and checkpoint inhibitors within that structure and perspective. But the broader vision is to say that is the direction the world is moving to in oncology is clearly from the one size fits all, the box therapy guidelines within the space. There are clear advantages of using standard technologies like simply measuring circulating tumor DNA as a marker for tumor dynamics over sometimes conventional biomarkers, even looking at imaging, where there's data which suggests even predating of some of those changes that can take place by weeks to months on the body fluids compared to what we find on specific aspects of the imaging portfolio. Despite the advantages of sequencing, one must be cognizant of some of the limitations that exist. Obviously, the challenges reside with regards to the sample acquisition, how you're preparing the samples in the lab, how that storage is taking place within the structure, how you're looking at different subsets of sequencing, whether it's for somatic single nucleotide variants, whether it's looking at small indels, various aspects of paired tumor DNA or copy number alteration reviews, and they're always important within this perspective. So as you can see within this slide, this was an excellent summary that came from Datar a couple of years back. Again, it's showing you not just looking at the genomic and the epigenomic, but what's really the microenvironment in play. And I think we've got some excellent speakers later this morning and tomorrow who are talking more about the microenvironment. In my view, that's absolutely critical to the next steps of how we're looking at this space. The blue and the red arrows that you see on the slide are just looking at the trends towards less tumor-derived targets or higher specificity or more tumor-derived with lower specificity for cell-free DNA as part of the spectrum. Okay, so this slide is just to showcase to you how the landscape is now changing also from the target perspective. Clearly, we are seeing new cell therapy lines being driven. A lot of that is very molecular derived from specific targets. Specifically, when we talk about the landscape that was driven a couple of years back and this data was from you, where we were clearly seeing a significant rise in terms of the active targets that were coming through from the new therapeutics, and the cell therapies has clearly gained the highest amount of mileage in terms of activity, and it's the second largest number of active trials after checkpoint inhibitors in the immuno-oncology space. And the immuno-oncology space clearly is driving a lot of enthusiasm That's been exemplified by a significant number of trials that are either underway or are getting going in the next few weeks and months in that space. So with regards to the impact, and this was an impact paper that we presented about a year and a half ago, or about a year ago, I think, which was really looking at what was going to be the impact on the cancer care ecosystem from the COVID perspective. And clearly, data that came out from the NCI estimated that a 1% increase in deaths from breast and colorectal cancer in the next 10 years, and equivalent would be in excess of 10,000 deaths just because of the COVID impact that would take place. Again, it may be an underestimate in terms of various models in terms of disruption of care, but similar studies that have looked at cancer diagnosis delays in the UK are already suggesting additional deaths in the range of between 5 and 15% across lung and colon and other cancer ecosystems in that space. So I mentioned a little bit about various aspects of genomic sequencing, which is the basics essence to how we're looking at large amount of smart genomics in this perspective. The next generation sequencing can be limited to a pre-specified group of genes, or can be even more broad-based. Looking at whole exome sequencing can involve the entire tumor genome if we're looking at the whole genome sequencing. And basically the choice of which approach one needs to look for is really going to be defined by what's the application, what is it that we want? Is it a clinical approach? Is it a research-based approach? What's the result that we're acquiring? What's the technical efficiency that we want to drive through that sequencing? And obviously all of them impact final aspects of cost. So the impact will be really from looking at the sample accrual to collection, to transportation to the lab, to interpreting the variants that exist. And this is really the pre-analytics, and this is really where those changes take place that help drive a lot of the differentiation in terms of the data false values, whether it's looking at the negatives or the positives within this space, and that they are critical to that drive and that process forward. There's been very little data till a couple of years ago that was looking at treatment strategies for relapse refractory metastatic cancers based upon comprehensive multi-analyte molecular analysis that would look at alignment to synchronous in vitro chemosensitivity in a label agnostic manner. So this is some data that we published in Oncotarget in 19. This was again a cohort of about 200 individuals where we were looking at label and organ agnostic treatment strategies for patients with those refractory relapsing metastatic cancers, minimal 2 lines of failure, some extensive lines of failure. And we looked at an integrated multi-analyte approach, something we called as an ETA, or an encyclopedic tumor analysis. And you can see the waterfall chart, which reflects Asperesis 1.1 criteria, how those patients benefited from some of those structures. So it's really trying to understand more about how tumors employ myriad mechanisms, feedback loops, and redundancies at each functional layer, whether it's at the coding, it's at the transcription, it's at the regulatory level. And what we did was we looked at analyzing not just the DNA, but looking at the DNA, the RNA, the cell-based activity, integrating that with pharmacogenetics to come up with treatment options that would have high efficacy and low toxicity profiling within that structure and help define that process. And in our view, using the encyclopedic approach of the DNA, RNA, and the cell together from a genomic standpoint, it really helps drive more of a capturing of and contextualizing data from multi-layered tumor interactome that helps that process. And again, you have various circulating biomarkers that represent promising non-invasive tools for that scenario and within the prognostication structure as well. So over the last two years, one of the areas of real interest which has been evolving has really been looking at cancer cells and cancer clusters. Traditionally, we've all looked at histopathology, looking at biopsy, an invasive procedure as a current standard to determine malignant status within those structures. Invasive biopsies do come where there are challenges, not to say in the least, which are anatomically bound, which may be challenging. though a lot of that has been circumvented by using interventional radiology. But then complications including pain, discomfort, bleeding, sepsis, at certain times looking at repeat biopsies across various spectrums and various structures, which may be difficult to manage. And through a blood-based approach of looking at clusters, so the clusters are basically defined as 2 or more individual circulating tumor cells that are bound together. But the clusters may contain additional traditional small or CK-derived circulating tumor cells that have specific biomarkers that help them identify as circulating tumor cells. And various studies have reported the presence of these clusters associated with metastatic risk and poor prognosis within the structure. In fact, one of the studies which involved prostate cancer showed the survival rates. Patients showed an 8-fold longer mean survival rate with only a single CTC vis-a-vis those who had clusters within the system. So the clusters do harbor the molecular and the functional imprints to look at those tumors. They are specific for various types of malignancy, and this is something that is of interest for us to look at in this space. Just to showcase you the clusters in terms of how they work. So hopefully you're looking at this video, which is running through. So you can see from the background of the initial cells, and I'll run it again. But effectively, you've got a wide microenvironment of various cells that are running, and this is a process that you're seeing over 4 or 5 days that's running through this process. And effectively, the cell and the clusters are coming together at the end of the process. So what you're able to see now are at the end those clusters that are being defined. We've done a lot of work over the last couple of years looking at these clusters from various subsets in one of the subsequent data points that I'm sharing with you. This was samples taken from about 5,000 cancer patients, which were stabilized and isolated for the clusters of the CTACs, which are the circulating ensemble of tumor-associated cells. And we found across those cells, we were able to identify in just close to just under 90% those cells across the epithelial organ malignancies. The interesting aspect of this process was also looking at trying to look at providing the CTACs that were detected in recently diagnosed therapy-naive patients as well within the structure. So which suggests the ability to identify a new systemic hallmark of cancer irrespective of what was the treatment status within the space. Increasingly, we've started to look at the clusters from various non-invasive diagnostic triage perspectives. So the objective of the study here that was run was really trying to look at these clusters from a diagnostic approach to try and define specifics within a tertiary cancer care center, whether it's possible to provide a non-invasive diagnosis for cancer with an accuracy that would not be inferior to the conventional tissue-based approach. That was one question that we wanted to ask. The second was whether they could be used for immunopathological characterization of tumors as per the tissue of origin, whether this approach would be suitable in real time to define the tumor dynamics in patients who've been pre-treated. And again, so just some of the data from the UK, where in about the 100,000 biopsies on the prostate cancer that happen, about 75%, 3/4 of those biopsies are negative, indicating a higher proportion of those who undergo the procedure may not have required it at that space and time. And is there a way to better have a conclusive diagnosis, which for the standard of care would still be obviously tissue-based, but if you could risk stratify those individuals by looking at a simple blood-based tool, as to those who need a biopsy tomorrow and those where the biopsies can be prolonged over a certain period of time to repeat over a period of within the next 6 months to 12 months, then that adds the benefit of trying to stagger and in an ecosystem. That was the interest in that perspective. We've looked at these clusters also in individuals who have been apparently asymptomatic within those cohorts. And we've actually found when we've followed up these patients who were initially asymptomatic in over 4,000 patients, patients who had the CTAC clusters had a 230-fold higher risk of a 1-year cancer when we followed up those individuals, and those individuals continue to be followed up. This was one of the initial studies we presented in cancer prevention last year, and some of that data is ongoing and on moving within this space. So I think the implementation of precision medicine through molecular profiling technologies, it's getting much more integrated, and we're starting to see much more of its role to try and enhance diagnostics, theranostics, and prognostication with an aim to improve clinical outcomes within this perspective. It's quite clear that we are moving into a greater utility of looking at omics, whether it's from the genetics, whether it's from other aspects, and looking at using them to look at the future of precision medicine, which will integrate a multi-omic tumor characterization, looking at dynamic monitoring of those individuals and patients through liquid biopsies using annotation that's automated in terms of disease structure. It's quite clear in my view that the work that's happening in genomics needs to be integrated with the clinician, the lab, to really provide real-time data points in this perspective as we move forward. Clearly, biomarkers have diagnostic, predictive, and prognostic approaches, and we're already seeing how tumor-agnostic approaches for the future of cancer with various sets of biomarkers, whether it's NTRK and others, where we've seen increasing approvals that have come through in that process. So again, cancer is it's a paradigmatic complex phenotype. It is complex in terms of the way it holds itself in that space. And I think what we need to understand is we need to be far more efficient in terms of looking at new ways to gather, analyze information that's layered together, that brings it together in terms of minimizing the caveats and complexities of the cancer under the domain of multi-omics. Look at it from a year and a half ago. We would have met at this meeting face to face. Travel would have been far easier. There would have been less struggle for resources. No desperation to cling on to mediocre evidence in terms of looking at what's best for the patient in terms of the outcomes. And when we knew that vaccinations were just for children, that's not the case anymore. The world does change, and we've got to change along with those. But I think with the changes that have happened with the pandemic, some of them have changed in terms of forever, in terms of the interface of where we are. We clearly need to understand that cancer is forever going to be a complex dynamic from a disease standpoint. It does change quickly. If we do need to clearly deliver the future of precision medicine, one that is reliable, that's robust, it's reproducible, it's ethical, where we're looking at diagnostics, stratification, treatment, monitoring, response, providing better outcomes, less futile treatments. That's all that we want for our patients. Then we've got a long way to go, but I think we're heading in the right direction. If anybody wants to reach out, that's my email where we can connect if there's any specifics. This is just the great people at our lab. We've got about 300 people at the lab working both in India and the UK and in Germany in terms of our teams that are working largely across those domains. I'm just going to end with what I always do, which is my favorite, which is I think we don't know it all, but to harbor that as the